फ्रेंड्स, स्वागत है आपका फिर से एग्ज़ाम क्रैक टीचिंग पर। हमने पहले 2 वीडियो में हरियाणा असिस्टेंट प्रोफेसर के क्वेश्चन 1 to 30 को सॉल्व किया था। आज के इस नए वीडियो में हम कंटिन्यू करेंगे 31 to 50। तो चलिए बिना टाइम वेस्ट किए शुरू करते हैं अपना अगला सेट ऑफ क्वेश्चन और देखते हैं क्या नया सीखने को मिलता है। लेट अस स्टार्ट फ्रॉम क्वेश्चन नंबर 31। Match the characteristic feature of given column A and with the species family given in column B. Male germ unit. This is a pseudo embryo sac, pollen grain embryo sac, and composite endosperm. This is very important. Composite endosperm, which is found in Hyacinthus orientalis. Pollen grain embryo sac, it is found in Podostamnaceae. Pseudo embryo sac, it is feature of Lorentheaceae. And male germ unit, the answer will be plumbago. So, male germ unit plumbago, pseudo embryo sac in Lorentheaceae, pollen grain embryo sac in Podostamnaceae, composite endosperm in Hyacinthus orientalis. Let us see first 4 B third. So the answer will be this. Okay, move to the next question number 32. Double fertilization was discovered in which of following plants? The question which came from this question, another important question is who discovered the double fertilization? So Nava Sachin in 1898 discovered double fertilization in Lilium and Fritillaria. So check करते हैं, yeah. So here it is the right answer. What happened? One male gamete fuse with egg and it give 2n zygote, and other second male gamete fuse with 2 polar nuclei that give 3 n endosperm. First male gamete, it is a first, it is a second male gamete. So 2n zygote and 3n endosperm का formation होता है. So this is the one of important. Now move to the next, that is question number 33. Entry of pollen through the funicles or through the integument is known as. This is very important question asked in many of exam. So please be careful whenever you do this type of questions. Always try to remember all way the pollen tube enter. The first is porogamy. Porogamy means, like, if I draw here, suppose this is the embryo sac structure. So with the pollen tube enter through the micropyder end. Suppose this is the pollen tube. When it enter through the micropyder pilar, and it is called porogamy. Next is chalezogamy. When pollen tube enter through the chaleza, here, if it is a chaleza part, then we called it as a chalezogamy. Porogamy, when enter through micropyle. Okay, कहाँ से एंट्री हो रही है? माइक्रोपाइल से. एंड चैलेजोगैमी थ्रू द चैलेजा, and through the micropyle. Now the next is mesogamy. When pollen tube enter through funicles or integument, we call mesogamy. Pseudogamy, when pollen tube, we will not talk about in pseudogamy about the pollen tube, but egg develop into embryo. When egg develop into embryo without fertilization. Okay, so this is pseudogamy. हमसे पूछा था entry of pollen grain through a funicles, तो answer will be mesogamy. Now move to the question number 34. If there are 50 microspore, please listen carefully. 15 microspore mother cell in anther and 18 of them degenerate. Okay, that means 50 megaspore mother cells हैं and 18 get degenerate. So we can say 32. 50 minus 18 is equal to 32 remain megaspore mother cell. Okay, the number of pollen grain in the anther would be, so pollen grain, you know, we will multiply 32 by 4 is equal to 128. So answer will be 128. Move to the question number 35. Match the type of meristem in column A with part of plant where it found. This is very interesting and very important. Lateral meristem, apical meristem and intercalary meristem. As you know, apical meristem will be at the apice region of a shoot and root. So here it is shorted. Now next, you all know lateral meristem. Lateral meristem found in the vascular and cork cambium region because it give a girth, and intercalary in the internode of leaf seed. So remember this 35. First ka third, here it will be the answer. Yeah, right. To the next question, question number 36. Here the column 1 and column B. In column A there is a term de-differentiation, redifferentiation and differentiation. First of all, we need to learn these terms. Okay, see, when we will discuss about the differentiation, simply in simple word I can say, when meristem change to permanent cell, so it is differentiation, meaning when normal meristematic cell mature into permanent cell or tissue for first time is called differentiation. Example, apical cell, yeah, apical meristematic cell when changed into root hair cell और xylem vessel, phloem, different तरह के permanent tissues बना रहा है, तो we will call it as a differentiation. Now second term है differentiation. जब भी हम डी डिफ्रेंशिएशन की बात करेंगे, दैट मीन व्हेन परमानेंट cell get changed to meristematic again, so we will call it de-differentiation. Now the third is re-differentiation, again name indicating. Those regained meristematic cell become new permanent cell or we can say after de-differentiation, those meristematic cells specialize again to perform a new function, like cork cambium make cork cell, like cork cambium make cork cell. It is example of de-differentiation. Now let us see here in the column. So de-differentiations mean loss of previous acquired character, obviously. Differentiations mean change in the form, structure, function and progeny of meristematic derivative and organization into new tissue. ठीक है? Okay, and redifferentiation, acquisition of new character. So the answer will be first ka second, B ka third and C. Yeah, so it will be the answer. The cork cambium is a good example of secondary meristem. Reason, it arises from the epidermis of various parenchymatous tissue in the cortex and deeper layer of bark. Right. So I think both the statements are correct, and it is correct explanation also. See, assertion A, cork cambium phellogen is a good example of secondary meristem, right? It is right because it form later and add secondary tissue. Reason, it arises from the epidermis. Correct. Explanation of how cork cambium originate. So cork cambium originate from the epidermis or various parenchymatous tissue in the cortex and deeper layer of bark. So it is right answer is this. Now move to the question number 38. Let us see question number 38. Given below the two statements, statement 1, the reaction center consists of chlorophyll molecules. It is right. Reaction center consists of a special chlorophyll A molecules, that is P680 in photosystem 2 and P700 in photosystem 2. A reaction center transfer a light excited photon from pigment molecules to molecules that use the trapped energy to drive chemical reaction. It is wrong. Actually what happen, लाइट हार्वेस्टिंग कॉम्प्लेक्स, जिसको हम एंटीना कॉम्प्लेक्स भी बोलते हैं, एंटीना कॉम्प्लेक्स. ओके, it contain accessory pigment that is like chlorophyll B, xanthophyll, carotenoid etc. These pigment, ये जो pigment है, these pigment absorb light. So suppose here the light. These pigment absorb the light, photons, and transfer excited energy. Transfer excited energy to reaction center. So the answer will be like this. So here we can say statement 1 is correct, but statement 2 is not correct. So the answer will be, it is, yes please. So the answer will be statement 1 is correct and statement, so answer will be the B. Now move to the question number 39. Water is required as a reactant in photosynthesis. Yes, it is correct. Water is required, as you know, water splitting होती है in photosynthesis में, so it is act as a reactant. The splitting of water provide electron for converting NADP to NADPH, which will be used to converting CO2 to sugar in Calvin cycle. Yeah, it's right. So we can say that both A and R are true, and R is correct explanation of A. It's very important. It's just topic from photosynthesis. Okay, photosynthesis, that is splitting of water होता है, and splitting of water से NADP convert होता है NADPH में, then which will be used to convert CO2 to ultimate sugar in Calvin cycle. Okay, now move to the next question number 40. The Calvin cycle function like sugar factory within the chloroplast. True, right? Because there is formation of a sugar, and using carbon from CO2 energy from ATP and high energy electron from NADPH, it construct an energy rich sugar molecules called glyceraldehyde 3-phosphate. It is, that is glyceraldehyde 3-phosphate की बात कर रहे हैं यहाँ पे, right? तो Calvin cycle act as a sugar factory in chloroplast, and it use CO2 plus ATP plus NADPH to make an energy rich sugar, that is, so it is true. Okay, let us move to the question number 41. Which of following products of reaction taking place in the thylakoids are consumed by the reaction occur in stroma? Listen, recall the process in thylakoid and stroma. When there is a photosynthesis process, there is a one organelle, that is thylakoid. You know, thylakoids and stroma are the part of chloroplast. In thylakoid, generally we call it as a site of light reaction, right? Also called light reaction or light dependent reaction. What happen in there? The key reaction happen, that is photolysis of water that produce oxygen, proton, and electrono. Okay, water gets split. Okay, light energy कल बताया था that how the energy get excite. Light energy excite electron used in electron transport chain to produce ATP and NADPH, right? So the product oxygen, ATP, NADPH in the thylakoid. Now move to the stroma. We can say site of Calvin cycle, that is light independent reaction. Light independent reaction. It use ATP, NADPH from light reaction to fix CO2 into sugar. So it requires ATP, NADPH and CO2. Now link product of thylakoid reaction to the stroma reaction. So what are ATP, NADPH are produced in the thylakoid membrane during the light reaction. ATP and NADPH then consumed in the stroma during Calvin cycle to convert CO2 into glucose or sugar. Oxygen produced in thylakoid is released, not used in Calvin cycle. Water is consumed in the thylakoid, not in stroma. ADP and NADP are regenerated after ATP and NADP are used in the stroma, but they are not product of thylakoid reaction. They are substrate recycled. So the answer will be water and so the answer will be ATP and NADPH. Water and H2O. अगर हमसे पूछा जाता water splitting in thylakoid, NADP positive, not. This is also not, and here it is not. So the answer will be NADPH and ATP. Simple, बहुत सिंपल था. दैट थाइलाकॉइड्स में लाइट रिएक्शन होता है जहाँ पे वाटर का स्प्लिटिंग होता है. ऑक्सीजन प्रोटोनों इलेक्ट्रॉनों बनते हैं जो कि फाइनली आपका ADP एंड NADPH की फॉर्मेशन करता है. देन इन स्ट्रोमा ये ATP और NADPH की हेल्प से कार्बन डाइऑक्साइड का फिक्सेशन होता है इन द फॉर्म ऑफ ग्लूकोज. Okay, so move to the next question number 42. Yes, in the what is in 42? Maximum AT production occur during which stage of cellular respiration? Maximum ATP production occur in, you know, it is electron transport. Okay, ऐसा क्यों होता है? The electron transport chain plus electron transport chain plus oxidative phosphorylation produce the bulk of ATP by chemiosmosis. Compared with, if we compare it with glycolysis or TCA. Okay, so the answer will be electron transport chain, electron transport, right? Now move to the question number 43. The number of ATP molecules produced from one molecules of glucose during fermentation is, that is the answer is simple. It's a 2. Fermentation regenerate NAD plus but yield only 2 ATP produced in glycolysis per glucose molecules per glucose. No additional ATP from fermentation step. So the number of ATP molecules produced from one molecule of glucose during fermentation is only a 2. Now move to the question number 44. Dash acid build up in human muscles during the strenuous activity और एक्सरसाइज. जब भी कोई स्ट्रेन होता है या हम एक्सरसाइज करते हैं, वी फील अ पेन इन दी muscles, right? So it's a very simple. I think it is a lactic acid. So the answer will be lactic acid. Now move to the next. Okay, one thing. यहाँ क्या होता है actually? The pyruvic acid convert to lactic acid. So accumulation in muscles. Okay. One more thing that now the question is ये pyruvic acid कहाँ से आया? तो under anaerobic condition muscles convert pyruvate. तो under anaerobic condition जब भी आएगी तो आपको पता है ग्लाइकोलिसिस से वहाँ पे पायरुविक एसिड का फॉर्मेशन पायरुवेट बनता है. तो वही पायरुविक एसिड लेटर ऑन जब ज़रूरत होगी एनाएरोबिक कंडीशन में तो वो लैक्टिक एसिड में कन्वर्ट होके स्टोर हो जाएगा एंड दिस विल बी द कॉज ऑफ पेन इन मसल्स. नाउ मूव टू द नेक्स्ट क्वेश्चन नंबर 45. The two plant hormone involved regulating seed dormancy and germination. Note down this, that the plant hormone या phyto hormone topic is very important. So यहाँ से आपको question आएंगे ही आएंगे. So always try to complete these topic. Okay. Okay. And now move to the question. So abscisic acid and gibberellin respectively. ऐसा क्यों? Because ABA promote and maintain dormancy. Promote, or we can say it maintain seed dormancy, right? Whereas another case, that is GA will do opposite. Gibberellins, what they do? Break dormancy. Okay. I hope. And then seed germinate. I hope the question is clear. The two plant hormone involved in regulating seed dormancy and germination for a certain period of a time. Seed in dormant condition, so that is due to ABA, and germination is due to breakdown of that dormancy, which is by a gibberellic acid, and the answer will be the abscisic acid and gibberellic acid respectively. Now move to the question number 46. Now move to the next 46. Which of following activity of soil bacteria does not contribute to creation of usable nitrogen for plant uses? Okay, तो इनमें से कौन सी एक्टिविटी है जो सॉइल बैक्टीरिया नहीं करते हैं, मतलब डज नॉट कंट्रीब्यूट टू क्रिएशन ऑफ यूजेबल नाइट्रोजन फॉर प्लांट यूज की यूजेबल प्लांट के लिए नाइट्रोजन नहीं बनता है. तो अमोनियम टू नाइट्रेट कन्वर्शन, व्हाट वी कॉल इट एज अ nitrofication, and you all know it occur by nitrosomonas or nitrobacter. Assembly of amino acid into protein. This is a part of protein synthesis. This is a part of protein synthesis inside organisms, okay, not bacterial soil contribution to a usable nitrogen. Move to next option, assembly of nitrogen fixation. Atmospheric nitrogen fixation, it is by rhizobium, azotobacter. Fix atmospheric nitrogen into ammonia, right, which is usable form. Formation of ammonium from protein in the dead leaf. This is ammonification actually. Fixation of ammonium from protein. Because formation of ammonium बन रहा है यहाँ पे, तो it means from protein, that is ammonification हो रही है, okay. So the right answer will be B, that is amino acid, assembly of amino acid into protein. That amino acid जो है वो protein बनाते हैं, उसको protein, it is a part of protein synthesis, not nitrogen fixations or nitrogen formation, okay. Now move to the next question, number 47. A non-allelic interaction where an allele of one gene has a overriding effect on the other gene. Overriding का मतलब हो गया, see, epistasis, ये एक तरह का क्या है, it is a non-allelic gene interaction. It is non-allelic gene interaction, okay. In this, one gene mask or modify the expression of another non-allelic gene. So the gene which mask the other gene is called epistatic gene, और जिसको मास्क किया जा रहा है, that is non-allelic genes, that is hypostatic gene, okay. Incomplete dominance, you know well, इसमें जैसे रेड मल्टीप्लाई वाइट क्रॉस करेंगे रेड वाइट फ्लावर का, हम पिंक फ्लावर बोलते थे, 1:2:1 रेशो आता था, तो दैट इज इन इनकम्प्लीट डोमिनेंस. दैट इज नॉट ड्यू टू द एनी मास्किंग इफेक्ट और ओवरराइडिंग इफेक्ट. दिस इज मास्किंग, ओके. सो द आंसर विल बी एपिस्टेटिक. कोडोमिनेंस का एग्जाम्पल आपका ब्लड ग्रुप हो जाता है. Multiple allelism is also AB blood group, ABO. Okay, now move to the question number 48, right? Now let's move. Here the 48. Polydactyly is a site of being an autosomal dominant human trait. It is an autosomal polydactyly. Trait is not frequently observed in the population because polydactyly gene has. Listen, polydactyly, in spite of being an autosomal dominant trait, is not frequently observed in the population. Otherwise, autosomal dominant trait can be frequent, but it not show in the population frequently. The reason, polydactyly show low penetrance, so not everyone with the allele show the phenotype, so it's a low penetration. We can say the answer will be the low penetration, okay, low penetrance. Now move to the question number 49. Which of following is an example of multiple allelism? I told you above that it is blood group ABO. 50 in C. In dihybrid crows, a true breeding parent with red and round seed. Red, it is dominant red and round seed. Sorry, it's a capital because round is a dominant character. Okay, in ना मेंडल में देखा था ना, the red and round seed was crossed with another true breeding parent. True breeding का मतलब homozygous recessive. Okay, here with a yellow and wrinkled seed. ये आ गया yellow and wrinkled and red and round, right? That means it is classical dihybrid क्रॉस दिया है आपको. ओके, जिसकी रेशियो कितने आती है 9:3:1. ठीक है, राइट. दैट इज इक्वल टू टोटल 16 इंडिविजुअल्स होते हैं ये. नाउ red is dominant over the yellow and round is dominant over the wrinkled. All F1 progeny were with red and round seed. On selfing F1 112 progeny were produced in F2. कितने? How many out of 1 on selfing? जब F1 की selfing की थी 112 progeny were produced in F2 में total जो progeny थी वो नंबर इतना था. Now how many of 112 progeny were red and round? तो ये जो रेड एंड राउंड है दैट इज डोमिनेंट. फर्स्ट वाला देखोगे आप, रेड एंड राउंड, ये रेड एंड राउंड, तो इट विल भी सबसे ज़्यादा होते हैं ये, इसलिए 9. तो हम क्या करेंगे 112 मल्टीप्लाई बाय 9/16. तो वी विल सॉल्व इट एंड जब आप इसको सॉल्व करेंगे द आंसर विल कम एज अ 63. So here the right answer is 63. Okay. All right, friends. That's for today's session. We have successfully solved question 31 to 50 together. I hope this was helpful and boosted your confidence. If you enjoyed the explanation, don't forget to like this video, share it with your friends, and of course subscribe to the channel if you have not already. Because in next video we will continue with the journey and solve the next set of the question. So stay tuned. It's going to be even more interesting. See you in the next one. Till then, keep learning, keep growing. Bye bye.